This focuses on the advantages and disadvantages of both RNAi and CRISPR, with a brief explanation of each. RNAi is a cellular mechanism in which naturally occurring double-stranded RNAs are used to silence the activity of specific genes. This biological machinery can be exploited to give an insight into a specific gene's function. In eukaryotes, the double-stranded RNAs involved can either be microRNAs or small interfering RNAs. However, due to the short nature of this video, there'll be a focus on siRNA. The first stage involves the designing and synthesis of siRNAs in a double-stranded form. Once successfully delivered to the target cells, dicer will cleave the double-stranded RNA into siRNA, which then combines with other proteins to form the RNA-induced silencing complex. This base pairs with the target mRNA, ending with cleavage and degradation of the target. CRISPR, like RNAi, is a naturally occurring mechanism that has been manipulated to allow precision editing of the genome. The most commonly used system is CRISPR-Cas9. Usually two different RNAs are needed, however they can be engineered into a singular guide RNA, the equivalent of the designed siRNA. The sgRNA combines with Cas9, a CRISPR-associated protein with nuclease activity, to form the effector complex. This will associate with a PAM in the target DNA, allowing Cas9 to make double-stranded cuts in the DNA. Either non-homologous end joining or homologous recombination will repair the break and in turn edit the target sequence. The reason for the exploration of these techniques is to decide on which to use for the manipulation of gene A in a eukaryote to get a deeper understanding of the gene's function. Although we assume both approaches work in the model organism, one disadvantage to RNAi is the issue of membrane passing. Depending on the cell type of gene A, there may be a need for an additional step incorporated to ensure that siRNA is still able to have an effect on the genome once inside the target cell. Other than this, if the siRNA is designed effectively so it has no off-target effects, as gene A only needs to be repressed, RNAi is a foolproof technique. CRISPR also has numerous pros and cons, many being very similar to RNAi, such as the off-target effects. However, it's more complex and thus involves many more stages. This can be seen as an advantage because more precise manipulation can be carried out. However, as the editing on gene A is rather simplistic, it means there's a greater room for error. Both techniques are similar in many respects, highlighted by this slide. However, RNAi for gene A is a better chosen technique. Although older, it is much more understood, having already been used to reduce blood cholesterol in non-human primates with no negative effects. As this technique needs to be able to reduce or eliminate the expression of gene A, RNAi is better suited as computer programs help design specific siRNAs to avoid off-target effects and eliminate gene A's expression. However, CRISPR is a much newer technique with many more ways in which it can edit a genome. Therefore, in the future when more is understood, or when given a different scenario, perhaps one in which donor DNA needs to be incorporated, CRISPR will be the desired technique.